So welcome everyone to our entomological club meeting. We have here Vladislav Ivanov speaking of the speciation process of Lycosidae spiders, which means the wolf spiders and etc. etc. And he has given his dissertation last Friday.
Yeah.
And I'm humbled and very grateful that he will be giving this presentation for us. I'm the president of the Entomological Club of Oulun, Harry Nystrom, and without further ado, here is Vladislav Ivanov. Thank you.
Hey, hello everybody. Thank you very much for coming on here to listen about wolf spiders. So mostly it will be about genetics, I think. A bit less maybe about spiders, but hopefully find it interesting. So let me just share my presentation. Great. Okay. Okay, so hopefully you'll see this. For those who have been here at the University of Oulun the previous Friday or watched the defense in Zoom, there will be quite much of a repetition. But also added some new slides that were not presented during the defense. Hopefully you find them also somehow interesting. So the topic of my work with past 7 years, I think, was the species delimitation and a little bit of systematics of wolf spiders, where I used two different methods of DNA sequencing, so which I will briefly introduce you. Why do we, in general, think about the species and how we approach it? So probably, as many of you have been collecting, and you understand that we do not have a proper agreement what species is, neither how we define them, how we approach ontological problem of what species is in general, nor epistemological problem of how we define the limits and what kind of methods are better. So this is the reason of this controversy and really many definitions of species. And it is important to somehow standardize and work so that we understand each other. We as scientists, we figure out that eventually the species is some kind of more or less universal unit, so we can communicate about research. And of course, it's important for all the policymakers because they need the standard units when they assess the resource we have. Because of course all the biodiversity we have around us consists of species of some kind of units, and according to current estimates, it's maybe 20% of all species are described. So we have a lot of work to do, and not so many people who actually can do that. So people suggested that we could standardize somehow species delimitation and species identification using DNA barcodes. And I think if you've been attending this club, Marco definitely talked about it, as well as Nina, as many others working here. So probably familiar, the DNA barcode is a single gene taken from mitochondrial DNA. And it has its advantages and disadvantages, of course. So, but also people work beyond, of course, DNA barcodes and try to find some kind of standards using some fractions of the genome, so sequencing not everything, but just part, or eventually analyzing whole genomes. And all of it, all this data, was used to somehow find these borders in between species or describe the speciation process, so we understand it better. And so we better define our species as standard units. So as I said, so barcoding initially identification tool. So we can just collect whatever, take one leg, sequence it, and know what it is without any kind of expertise in particular group of organisms, because we already have quite large databases, which is cool. But also it was used as additional line of evidence for species delimitation. So if we looking for borders, we actually do not know, if we collected some specimens of, say, flies or spiders. We need to figure out if they're the same species or not. We could use this DNA barcoding thing, but it has, of course, limited resolution. So in spiders in general, in the world, like there are already almost 120,000 DNA barcodes. So people have been working, of course. So we know some patterns of genetics of this particular single gene in spiders, but of course it's quite biased. In Europe, for instance, it's only 10% of the fauna like that has this DNA barcode. 44%, 10% of all these breeds, like they come from Europe. So 44% they're coming from Canada, meaning that the center of research is actually there. So it doesn't mean anything else, I think. 8% from US, probably because of the proximity. And actually the most diverse places are not yet sampled sufficiently, because out of 120,000 sequences, only 38% they come from all the tropics, so all the nice places like Australia, Southeast Asia, like and Amazonian forest. Even more so, like 20% of European spider fauna is barcoded at the moment. So there's still a lot of work to do, and hopefully it will be accomplished at some point. But what I find interesting, this is probably a graph that it's hard to comprehend at the very first sight, but because it combines data from Europe, like European Union, like it combines data from Canada, and it shows at the bottom interspecific distances or within species distances. So it's how different each particular specimen within species. It's like how I am different from hyönteiskerhon, apart from having longer beard. Like, so genetically we should be quite close, I think. So this is the distribution of these values, like so. And this would be, if we continue analogy, like the distance to say chimpanzee, like, so this would be like our nearest neighbor that we have, right? But this is for spiders. And we have families, quite many, familiar to those who have been collecting them here. And we see that, patterns here in Europe of these distances in DNA barcodes, and in Canada with different species, they are kind of the same. So actually what it shows is that there is a high chance that whatever is kind of above this, some, let's say 5% threshold, like a difference, like would be a different species, like something like that. Like, but there are groups where these distances are kind of shorter in between species. So somehow we are closer to bonobo than we would love to be. And one of these families is Lycosidae. Some other families, this is outlier clearly, like so, this Miturgidae. But most of the families, the distances between defined species is quite large, like say on average. In Lycosidae, it's shorter, like, and we have even more problems, where we know that there are different species, right? Like, so they were defined quite clearly by people working like Kronestad, for instance, like that. My groups were largely described or properly delimited by Kronestad and Oulun from Sweden. And it's a mess. So it should be that each particular cluster, like of these names of this Protivagus fagnicola species, they should be like different branches, like so different kind of small bushes there. And the same should be for Apicosaculeata and Teniata, but they are kind of mixed. Like, so see, this single gene, this DNA barcode, clearly cannot recognize between species that are recognized by morphology, recognized by ecology, behavior, and a lot of work done by classical taxonomy. So having this problem is interesting. Like, why is that? So in my work, I used so-called DDRAD sequencing. Technically, I'm just selecting some fraction of the genome and sequence it. It's millions of base pairs, so it's 100,000 times longer sequences, or 100 times more information than the single gene. So, and I hope that I kind of achieve this greater resolution. So double digest red sac, Lycosidae did the red sac, what I'm actually doing, so I extract the DNA, right? So this is our representation of our DNA. I chop it into pieces, like and specific restriction enzymes from different individuals, and I have this very short reads that I can compare and get my data. So that's the idea. So I have 1-2% of the genome, like, and I'm just trying to again build trees, like see how different species are, like, or what we expect to be different species. And this is methodology, and in terms of the actual studies, I picked some of their Lycosidae because first it's this wolf spider is easier to collect a little bit. So you can put the pitfall traps and you're happy you get large numbers. And again, because of their barcoding patterns and because they are so morphologically almost distinguishable in many cases, like females, for instance. And then I picked different geographical settings. So I was collecting like in symmetry, like in Finland. So here, going in Farmin, like Kilpisjärvi and so on. So collecting my beautiful animals. And then I compared some other studies with some other species with their counterparts in even North America, in Far East Russia. So wider geography was sampled. And I look at the patterns. So one of the example cases I have, like this, that what I showed in the trees, is Fulvipes protivagus fagnicula ripare and polata. Like, so of course, if you were identifying and Arikakka present here, of course, recognizes them very nicely. Like, so it's not a problem for him. But of course, they are quite uniform, like so these guys. And I made some mistakes with my identification at some point. So, and again, as I showed in one of the slides, this is again another representation of this mess, like where a single gene cannot distinguish in between different species. So the idea is that we should have these colorful circles not to be mixed up and not to be so close to each other. So this would be normal situation to have these longer distances. But when I use my DDRAD sec, like so the mass time using this fraction of the genome, I have these clusters corresponding very, very nicely to the actual species recognized by all the classical methods. It's really cool. Like, so meaning it's working, like, and even more so, it sees in symmetry. So they're kind of more or less from the same locations, meaning that the method kind of picks up the variation observed with other methods. Like, so why do we have then this kind of strange pattern like in the single gene? So it's mitochondria, it has its problems, like so, and of course could be talked a lot about. But what we suspect that at some point, like in the past, like some part of the genetic material in a way was mixed up in between species, like, and we tried even to estimate how much. Like, so and some points when the species were just separating from each other, they were young, like, so they still could mate, like, and somehow they exchanged some of the genetic material, like, and one part of the genetic material was mitochondrial DNA that just became introgressed or fully spread among all the species. So this is kind of the case in symmetry. So now we understand that, okay, we have an idea that this could be the reason. Some older admixture is kind of similar to what we have with Neanderthals. Like, so, and methods that we're using, like, they're similar. So you probably heard about this 1-2% in all Europeans DNA coming from Neanderthals. Like, so it's the thing. So it's like interspecific hybridization or introgression. So here it's like similar patterns, and that we think explains this mitochondrial DNA strange behavior, this DNA barcode strange behavior. Then I picked another species, like not comparing them to each other, but taking species of Pardosa ripare from Finland and from actually Far East Russia, like it's like almost the Pacific coast, like near Magadan. And also apply the same methods, like so. So two species where I tested how different. So now we expect that this species, say Pardosa ripare, is spread all the way from Atlantics to Pacifics, right? And it's just one species. So I kind of collected specimens from the edges of this distribution, like and compared them to each other to see if they are really the same species or not. Because we know that DNA barcoding can be misleading. Like so, I applied the methods like DDRAD and also morphology. So I did some measurements, and I did the same for Pardosa palustris. So again, so back to the system, like of representation of the single gene patterns. For instance, for Pardosa palustris, the single genes is single DNA barcode pattern is that the specimens are very close. They're not really different. But we know that it can be misleading again. So as in Lycosidae, might be a problem. So we did morphology method.
Sorry.
Yes.
It seems that the slides are not changing in the presentation online.
How far should I go back?
They are seeing the first slide.
You should start from the beginning.
Okay, so great, like
you can skip through the slides very fast. Yeah, you're sharing the desktop instead of the.
Okay. So, all right, that's very weird because I just shared the presentation, like so. How come? Yeah, that's what I did. That's exactly what I did. And
go to the slideshow.
All right. Okay. So let me think. Okay. So do you see anything? So if I can anybody confirm that they're seeing anything now? What do people see?
I see the slide 18.
Good, that's good. Okay. Is it still visible, like and understandable if I just keep it like this, or still can try, of course, just showing it like this. So what do you see now?
Now I see the first slide.
Only the first slide.
Yeah, but go to the next one.
And now the second.
Not yet.
So
No.
it's the first slide.
Just don't make the slideshow.
Yeah, if you first go to the presentation mode and then you select what you share. You share the presentation. Can you sharing?
Should I stop share?
Yeah.
Okay, and share again.
Yep.
Okay, that's weird.
Oh, it doesn't. Yeah. Yeah.
So this one and right. So do you see the change?
Now it changed. Yes.
Okay,
Excellent.
so just yeah, it's the same problem. We didn't recognize it's a second screen for this computer. Like, so sorry about that. Like so, it's
Yeah. Sorry that I was just listening. I didn't notice this before.
Thank you for noting. Yeah, right. Okay, so as I said, so I probably just briefly repeat that, you know, the species problem is still a problem, and we cannot avoid it, like so, because we cannot figure out the proper parameters yet, but we try to standardize it.
You might have to go to talk to
Right.
Again, I think they were listening, and I think that.
So yeah, this is the numbers of spiders like barcodes. So probably if you're interested, like, so that's how much we have from around the world. So there's a lot to be done again. And this is a slide where I was talking. So that DNA barcodes actually discriminate quite nicely spiders, what we believe to be different species from within species variation. Like, but in some families you have a problem. So where it is actually an average lower, like in wolf spiders. And that, as I was showing more of these trees, I can so on. So the DNA barcodes can be misleading. So on the tree, it would look like a mixture of things that we believe to be different species, but they are quite much together. right? And then again, that's how I picked my genomic data. This is a target group. That was one of the target groups where we established that some kind of introgression was occurring in between this really nicely defined species and nicely defined by the genomic data, but still not by the DNA barcodes. And here is a slide where we could estimate somehow like the amount of admixed DNA in between species. And I think I have reached a point where I talked about species with a wider distribution. So 2 cases analyzed in exactly the same way, where I tried to figure out what is the patterns of DNA barcodes. And again, so we believe it to be the same species. DNA barcodes say, oh, it is the same species. Nice. Kind of should be working fine, but we know that DNA barcodes not necessarily work in this group. So I added morphology and for both species, like, and the blue dots, like they coming from Finland, the yellow ones from Far East Russia, like, and it seems to be that even if we do classical morphological approach, just measuring like all the legs, all the things, we still cannot really discriminating between these two really distant populations separated by thousands of kilometers, except for females, like somehow in case of Pardosa seem to be more clearly separated, but yeah, indeed they're kind of larger. And again, so our genomic data, it picks up this finer structure like of populations, probably, like, so it thinks that you know, population from Far East Russia and from Finland, they actually separate quite nicely. So our genomic data suggests that they are really different, right? For this, for Pardosa riparia case, and for Pardosa palustris, again, the tree suggests that they are different. So if I only had the tree, I would have been forced to say that species in Finland is separate, like, from the species that we find in Far East Russia. But then, of course, we add more data, like, and more analysis, like with the same data, and then it becomes problematic. Like here, for instance, it all merged together. So these colors correspond to kind of population or species. Like if there are different colors, there are different species. So it's kind of the same data, but different analysis, and different analysis suggest different things. So eventually what I learned from here, like what I picked up, is that this is exactly the case where you hit the actual evolution at the crime scene. Like, so the spiders are, these guys, they are kind of diverging. So it's like new species forming. That's exactly where I caught them. Like, so, and it's very nice to study in future because of course we'll know what kind of factors will influence like the formation of new species. Like so. But at the moment, we could have been saying that it is like a subspecies in Far East Russia or something, but spider tradition doesn't like subspecies. Like, so I know the butterflies, there are so many, like it's just crazy. Like, so spider people decided, nope, like it's a bad idea. Like, so you were sure or not, like, so. And then the third case, which is kind of interesting, it's more complicated. So I kind of went with increasing of geographic complexity. So there's this kind of typical looking also Pardosa from so-called Pardosa Lycosidae group. I have specimens where I have sequenced genome, like data, so some part of the genome, like so marked with stars. And also, of course, this single DNA barcode, so one single gene, like, and also have some samples where you have only DNA barcodes. So what we actually have, so we have so-called Pardosa hyperbarea, quite massive species, like also here, like in bogs, like and farther in the north, like so quite familiar to spider people. So there is Pardosa areophila, like, and it should be in Alps, like, and Pyrenees, more or less. So what I actually observed that in Canada this Pardoza hyperbarea, which is believed to be spread all over Europe up to Ural Mountains, maybe a little bit in the West Siberia, and all through Canada. So even the single gene kind of sees this Pardoza hyperbarea as complex of 3 species, like so. One in Finland, like so, or say in Europe, like and to the east, and 2 in North America. Then the problem arises is that the DNA barcode, this single gene from Pardoza hyperbarea, again is shared with another species, like so, but in Europe, that occurs in Tatra Mountains, like in Slovakia. So making it a mess, like so. So simple solution would be like, okay, merge these 2 like as one species, describe these 2 as 2 new species. And then this Pardoza reaphylla case is more complicated because I don't have enough samples from Alps, unfortunately. But then you do some analysis with this DNA barcodes again, like so. That's where this mixture is shown very nicely, like so. All yellow is supposed to be Pardoza hyperbarea, right? Like so, European Pardoza hyperbarea is mixed up with Alps and Tatra Mountains, like so. So technically, what I have here looks very much on DNA level, like of the single gene. It's very much like something I find in the Alps, but not in Pyrenees. So in Pyrenees, like so, everything is fine, like so. It's really distant, as you can see. And in Canada, it's 2 clusters, like so, occurring in perfect symmetry, like just in one spot, or like so, in type place, like where it's kind of cool. So again, it's mitochondrial DNA tree. Like, but if I do my genomic data, of course, like so, of course, I pick up difference between Pardoza hyperbarea from Finland and Canada and Greenland, obviously, like so, it was observed. But then this complicated case where DNA barcode here is shared between Pardoza hyperbarea and Saltuaria from Slovakia, it kind of disappears. So Slovakian population is still kind of valid, like it's as a separate species, it's no problem with that. Like, and again, Pyrenees like something really, really different. So what happens here is a mess, like so. Very likely it's linked to some glaciation process, and I did some this introgression tests of like in the first case, and there seems to be some of that mixture in between Pardoza hyperbarea from Finland and Saltuaria and even Oreaphylla, like so. There seemed to be the old exchange of genomic material that led to spread of one type of mitochondrial DNA in Europe. But in Canada, like of course, like this is split quite a long time ago, it actually diverged so much that we could actually speak about 2 different species in Canada. So what I figure from this is that the species level, like so, this genomic approach works really, really nicely. And then I tried to kind of push it a little bit beyond and just talk about a bit of systematics of things. So there is genus Pardoza, and all of them here, like except for 2, like are kind of Pardozas. If I use my genomic method, like thingy, it seems to be that at least 2 genera, like are very much within Pardoza, like so. And then there comes a question. So what you recognize as Acantholicosa or Pyrenicosa should be synonymized with Pardoza, like, and it has been a matter of debate, I'm afraid, for 100 years now, like so. And it seems to be that this kind of reduced genome representation is 2% of the genome, like is enough to have some kind of conclusion, but it's not published yet. And it's not so easy, But anyways, like this kind of genomic data can recognize so-called species groups that could be also potentially split into different genera, like so. So actually, there we have 2 choices: whether synonymize everything that looks like Pardoza to Pardoza and kind of continue having it as a garbage bin, or increase our sampling and do a lot of work, like and split Pardoza into many different genera. Like so. But it's kind of good news because the method is cheaper than the whole genomes, like it has its disadvantages, but on the level of apparently subfamilies, like so it works very nice, probably irrespective of the taxon, which is kind of cool. So yeah, so those were my major conclusions. So we can use DRSEC equally, like with equal success as traditional approaches. So this allopathic, like distant populations are problematic, and DNA barcode DRSEC discordance largely explained by many historical factors. So there could be historical exchange of the DNA, and probably it will be more common in almost all the groups. And again, for nice phylogenies, we still can use DRSEC, but probably not for the deep one. And thank you so much. So, and yeah.
Question about this general.
Yeah.
Is the synonymizing a lot of them or splitting it up?
No. Right.
So that are they if you split this part also into many, because it's not monophyletic, split in many genera,
right,
it makes it doesn't